Why is predicting CFTR gene mutation outcomes so hard? Imagine trying to forecast exactly how a single change in a gene will impact a person's health. That's what makes predicting outcomes of CFTR gene mutations in cystic fibrosis so complex. Cystic fibrosis is caused by mutations in the CFTR gene, which makes a protein that controls how salt and water move in and out of cells. This process is vital for healthy lungs, digestion, and other organs. But the tricky part is that not all mutations affect the gene or the protein in the same way, which makes predicting how severe the disease will be very challenging. The CFTR gene is quite large and has thousands of known mutations. Some mutations stop the gene from making any CFTR protein at all. Others cause the protein to be made but misfolded or non-functional. Some mutations change how the protein is regulated or how long it stays active in the cell. Because these mutations impact different parts of the gene and protein, their effects can vary widely. For example, a mutation that prevents the protein from being made will likely cause more severe symptoms than one that only slightly reduces its activity. But even mutations that seem similar can have different effects because of how they interact with the protein's structure. Advanced techniques like molecular modeling help scientists understand how specific mutations destabilize the CFTR protein. Some mutations cause the protein to fold incorrectly, leading to instability and malfunction. This instability can disrupt normal cell processes in ways that are hard to predict just by looking at the DNA sequence. Even small changes in the gene can have unpredictable effects on how well the protein works, making it difficult to determine the exact outcome for each person. Adding to the complexity, the environment and other genes influence how a mutation expresses itself. Two people with the same CFTR mutation might have very different symptoms or disease severity because of differences in their overall genetic makeup or environmental exposures. Factors like infections, nutrition, and lifestyle can all modify how cystic fibrosis manifests, making predictions even more uncertain. While genetic testing has become more advanced with high-throughput DNA sequencing, interpreting what these mutations mean for an individual remains difficult. Some mutations are clearly linked to severe disease, but many are of uncertain significance. Different prediction tools can give conflicting results, which complicates counseling and treatment decisions. This means that even with detailed genetic information, predicting exactly how cystic fibrosis will develop in a person is still a challenge. Another layer of difficulty is that scientists don't yet know precisely how much functional CFTR protein is needed to keep someone healthy. Some research suggests that even a small amount of normal CFTR activity can improve lung function, but this threshold varies from person to person and organ to organ. Because of this variability, it's hard to say how much a treatment needs to restore CFTR function to make a real difference. In summary, predicting the outcomes of CFTR gene mutations is hard because of the gene's complexity, the wide variety of mutations, the influence of other genes and the environment, and the limitations of current prediction methods. While ongoing research and technological advances are helping us understand more, much remains uncertain. This highlights the importance of personalized medical care and the need for continued research to better predict and treat cystic fibrosis. If you or someone you know is affected, always consult healthcare professionals for tailored advice.